Today we'll be talking about simple diffusion, facilitated diffusion, and active transport. Simple diffusion is the diffusion of small, non-polar ions directly across the membrane. Polar molecules are unable to cross the cell membrane through simple diffusion due to the hydrophobic interior of the bilayer. Molecules diffuse from areas of high concentration to areas of low concentration. Simple diffusion is the diffusion without the use of transmembrane proteins or ATP. Molecules such as oxygen and carbon dioxide are able to diffuse through the membrane through simple diffusion. Facilitated diffusion is the diffusion of polar molecules and molecules too large to pass directly through the bilayer. Molecules cross the membrane with the assistance of transmembrane proteins embedded within the phospholipid bilayer. Molecules diffuse from areas of high concentration to areas of low concentration. Molecules pass through protein channels, which allow molecules directly into the cell. Molecules can also enter carrier proteins. When a molecule enters a carrier protein, it causes its shape change, allowing a molecule to enter the cell. Active transport is the diffusion of molecules against the concentration gradient with the use of ATP. A common example of active transport is the sodium-potassium pump, which is used in cells to regulate the concentration of sodium and potassium within the cell. Notice that the concentration of sodium inside the cell is lower than the concentration outside the cell. In addition, the concentration of potassium is higher inside the cell than it is outside the cell. Let's take a look at how it works. Active transport begins with 3 sodium ions inside the cell bind to the sodium-potassium pump due to its high affinity for sodium ions. 3 sodium ions bind to the pump and initiates a phosphorylation with the use of ATP. The phosphorylation allows the pump to change shape, lowering its affinity for sodium ions. This allows the ions to be released into the extracellular fluid. The change in shape triggers a high affinity for potassium ions, allowing 2 to enter the pump. This triggers the phosphate group received from ATP to be released. When the phosphate group is released, the sodium-potassium pump loses its affinity for potassium ions and changes shape once more. This allows for the release of the potassium ions into the cell. The pump now has an affinity for sodium ions, repeating the cycle. The molecules are able to be pumped against their gradient with the use of ATP.